This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 181, recorded on Friday, April 27th, 2012. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hi there, Vincent. How are you today?
Very well, Rich. The beautiful day, and the shuttle, not too long ago, just flew by.
Was that just today?
It was this morning.
What's it? I thought it was already in D.C., or is this a different one?
I think, I don't know. It may have come here.
Because one flew to D.C. like last week or something.
Okay, so this is a different one. This is the Enterprise.
I guess the shuttles are being dispersed.
That's right. To all the politically forward cities in the country.
I guess they won't make it here.
Yeah, so it's on its way to the museum on the Hudson, the Intrepid.
Okay, yeah, so that must be a different one. Because
anyway,
there was one that went to the Smithsonian.
Yeah, it's a different one.
Yeah.
It's really cool. It just flew right by my window.
Yeah, I saw the video.
I put a video up. So that's pretty cool. That doesn't happen every day, so it's a pretty good day. Also joining us today, returning from the University of Michigan, Kathy Spindler.
Hi, Vincent. Hi, Rich.
Hi, Kathy.
So you decided to come back because you liked it last time?
Yeah, it's a lot of fun.
It's your third time, actually.
It is.
It could be habit forming.
Yeah.
Everything been well with you out there in the Midwest?
It has.
Did you have a rough winter or no?
No, we had the mild winter that everybody had. Although this morning I decided on my bike I would just wear my winter coat. It was 31, and rather than do several layers of coats and have to figure out which ones were the right combination, I just wore the winter coat.
31F, you mean?
Yes, 31F.
Oh,
it's pretty cold, man.
Yeah, well, it's now up to 50.
Rich is in the 80s down there, right?
I got 83 Fahrenheit, 28.33 Celsius.
I can't see why anyone lives in Michigan.
Well, it's 50 now. It's gorgeous. It's 50, and that's 10 Celsius.
Yeah, I'm just kidding.
So, yeah,
soon it'll be. So what is the hottest it ever gets out there?
Oh, it gets in the 90s.
Yeah.
For not very long periods of time. But I lived in Georgia, so the 90s doesn't seem too bad to me.
Yeah, that's right. That was really hot down there, right?
Yes, kind of like Rich's Florida.
Yeah.
But still cool, but still cooler than Florida. We had someone on, we had Trudy on a couple of weeks ago in Georgia, and her temperature was substantially less than Rich's on that nice day.
Yeah.
Let's see what it is here in New York City.
I was going to ask.
It is, it's 10 degrees Celsius. It's a little chilly, isn't it?
What does that mean?
It's the same as here, yeah.
I got to look it up.
It's the exact same as here. It's 50 degrees Fahrenheit.
Yeah.
Is that right?
Yeah, we had a warm spell, and now it's just cold again. It doesn't freeze or anything, but it's chilly. It gets down into the single digits at night.
Wow.
So there you go. Anyway, it's a nice day here.
I'll take 83.
Apparently. So did you chill last weekend, Rich? Is that okay?
Yeah, we got a little chill last weekend, but nothing serious.
No, did you chill? You were going to chill.
Oh, did I chill?
Yeah, you said. I said, what are you doing last weekend?
The weather got cool. Yes, I did chill. I did nothing. I read a novel.
Good.
Watched some movies. Nothing.
All right, good. I it's important that you chill like the Floridians do.
Yeah, right.
Okay. Huh. All right, we have a couple of really cool stories today, as usual, but I thought we had a bunch of email with respect to the last episode, so I thought we'd do those first just while the last episode is fresh in everyone's mind. Is that okay with everyone?
Yeah, sure. Great. I like these follow-ups.
One is from Horatio. He actually posted this on the website. A brief comment on rabies. The genotype 1 rabies virus usually sustains itself in an endemic cycle in different carnivores, according to geography and species composition. Bats may carry genotype 1, but are not needed as reservoirs. However, they are reservoirs of the specific bat genotypes, which may cause disease in various animals and humans. Fortunately, the basic reproduction number of rabies virus is low, primarily due to the particular mode of transmission, restricted tissue tropism, and long incubation time. Let's hope that the path to a putative, aerogenically transmitted virus is long and with many obstacles. That's a great statement. I like that.
Right. So we had we had some sort of discussion about this last time where there was some uncertainty about what the real reservoir was and that kind of stuff. And this basically reflects what I was thinking was the case, which I'm glad is right, because I looked this up. I was curious about this and looked it up a long time ago because I teach this stuff to the medical students and I want to get it right. And I also looked it up in fields and got basically the same thing. It's weird because it's has a very broad host range, and you can't really say this is the reservoir. Okay, it's passed around in whatever happens to be the dominant species, and that's mostly dogs. Domesticated dogs, well, semi-domesticated, feral dogs. The only reason it's not a problem here in the U.S. is because we are very vigilant in screening and vaccinating dogs and pets. So here the dominant rabies-carrying species is raccoons. They're wild animals. But elsewhere, in particular in places like India, dogs are a big problem.
Yeah, that's a good answer. Isn't TWiV a learning experience?
It is indeed.
It's not just us doing it; it's our listeners. They chime in. It's great,
right? And we listen. We learn too.
If I could only remember all this stuff more than a month.
You do pretty well. You come up with you do pretty well.
Yeah, I'm always impressed with the things that you can recall. So.
Our next comment is from Erica. Hi, Team TWiV. Hey, we have to get shirts that say Team TWiV on them, right? Wear them while we do the podcast. Oh, that would be really nerdy, right? As if we're not already. Right. I just wanted to write in and let you know that you did indeed discuss MIR-122 and HCV in a previous podcast. As Rich suspected, it was episode 97 with my PI, Peter Sarno. Not only is Peter a fantastic mentor, but he is a pioneer in the field of virology, and his episode on your show is one of my favorites. I highly recommend it to anyone who is interested in learning more about MIR-122 and HCV. Thanks so much for a great podcast. I was out there and I interviewed Peter, and I forgot that we had done that. There you go.
Right.
See?
Yeah, you forgot.
That was a dual show with Peter and Bert Semler.
That's right.
California Virology.
Right.
Yes, I remember that. The next one is from Rohit. I was pleasantly surprised to hear our MIR-122 and HCV paper discussed on TWiV 180. We are really glad to know that the TWiV crew liked our study. Did you get a TWiV bump, man? Thanks for discussing our paper. Currently, I'm a postdoc with Dr. Benjamin Tenover in the Department of Microbiology at Mount Sinai, where I'm trying to further develop the novel technology of use of host miRNAs to engineer live attenuated influenza vaccine. This technology was demonstrated earlier by Jasmine Perez in the Tenover lab, and he provides a reference for that. I did my PhD with Dr. Stan Lemon when he was at UTexas Galveston. Some of the initial experiments of the PNAS manuscript were actually carried out in the Lemon lab when we were located on the 5th floor of the Galveston National Lab, where you are headed sometime soon to do a TWiV episode. Yeah, that's right. You know, Rich, we have to arrange that. Remember, they've been trying to get us for over a year.
That's right.
Maybe we could combine it with the Austin trip.
Yeah,
although
that's
probably
an idea.
They're as far away as you and I are, right?
I don't think it's that bad.
I thought that I would answer some of the questions that came up during the discussion of the paper. In addition to its positive role in HCV replication, MIR-122 also regulates cholesterol metabolism in liver. Knockdown of MIR-122 significantly reduces serum cholesterol levels in mice as well as non-human primates. Thus, MIR-122 is also an attractive drug target for cardiovascular diseases. However, certain liver cancers are associated with lower MIR-122 levels, suggesting that long-term knockdown might have some side effects. Location of MIR targets in 5' UTR by itself is not enough to change a MIR from a repressor to an activator of gene expression, as there are examples where MIR targets located in the 5' UTR of mRNAs downregulate gene expression. Thus, miR-122 effect on HCV 5' UTR seems to be unique so far. However, there must be other such examples out there that are yet to be discovered. Allen was right in pointing out that RISC, as the name RNA-induced silencing complex suggests, is a complex of multiple types of proteins. Argonauts are the central and canonical components of RISC. There are 4 Argonaut proteins in the human genome, Argonaut 1 to 4. Argonaut 2 is the best studied one and is the only Argonaut with a slicer/endonuclease activity that is used to cleave the target mRNA when there is perfect complementarity between miRNA/siRNA and its target. You can tell this guy did his PhD on microRNAs, right?
Yeah.
Definitely.
Good job. Sorry. Good job, Rohit. Thanks for the info.
Yep. And
Go ahead.
It's a 4-hour drive from Austin to Galveston.
Yeah, we could do that, right? It's about the same as from
Gainesville to
Florida.
where did
Gulf
we go?
Coast, yeah.
Fort Myers.
Yeah.
We won't have the TWiV mobile though.
You would probably fly to Austin, right?
Yes.
But we should rent a car and drive to the other place, right?
Sure, why not?
We'll figure it out.
That gives us the opportunity to, what we were going to get, a canary yellow Mustang?
Yeah.
Ooh.
Convertible.
Yeah.
I'd like to do that. All right. One more follow-up from Steven. I'm a new subscriber to TWiV, and I just began with number 180. I think he means 179, actually. Rich Condit's comments on polydnaviruses and wasps was fascinating, and I will follow up on that. A few comments. First is a broken link. 2, braconid wasps are parasitoids, not parasites. Parasitoids kill the host.
Okay.
Next, the layers of complexity involving parasitoid wasps are sometimes even deeper. Here's a photo I took by accident while shooting some grasshoppers I casually study in the Olympic Mountains. I've appended a comment from bugguy.net to which I contributed a crop of this photo. So he's got a picture of 2 grasshoppers, and on one of them there's a wasp sitting on near the head. It's beautiful. These are hyperparasitoids of common grasshoppers, though sarcophid and tachinid flies, more commonly found in western states. Great find. The flies parasitize or parasitoidize the grasshoppers. Then the wasps lay an egg on the grasshopper. The larva of the wasp then finds the larva of the fly and parasitoidizes it. Amazing. I have to wonder how much of a role viral genes play in all of this.
Really?
It's amazing.
It's crazy.
I had no idea that this went on.
It is just crazy out there. And this is an accidental shot because those there are actually online. There are videos of these wasps laying their eggs in the caterpillars, and it's really quick.
Mm-hmm.
So
yeah, he said like 1 second or something.
Yeah, so it has to be he was just trying to take pictures of the grasshoppers, and the fly got in there.
So I guess they're mating, right?
Those 2 grasshoppers are, yes, indeed.
Great, you're mating and a bloody wasp comes by.
Right. Like,
there's no peace in the world, right?
Yeah. You're trying to wreck my day here.
But it is so cool that the wasp is somehow, the eggs are destined for the fly, not the grasshopper, right?
Right.
Right.
Yes.
And if you looked, you'd find viruses involved in that. It's
Probably.
Just amazing, or whatever you want to call them.
Yeah.
Okay, thanks for that. Now we can move on to our papers. First paper is just published recently in the Journal of Virology. We don't do a lot of JV papers. I guess they're a little more complicated. It is called Biological Characterization and Next Generation Genome Sequencing of the Unclassified Cotia virus, SPAN232, poxviruses. Do you know how to say that, Rich? Cotia?
Cotia.
Cotia?
Mm-hmm.
It's named after some place in Brazil, right?
It's named after a town just to the west of Sao Paulo.
São Paulo.
Yeah, SAO, however you pronounce that.
That's what I learned that when I went there last year.
That's right.
So the first
actually, a Cotia in Portuguese, a Cotia is also the name of an animal.
Is that right?
Which is in I think it's in agouti.
Oh yeah.
So when you talk about Cotia virus in Brazil, it gets a little confusing because they think it's a virus of this animal, but no, it's just named because of where it was isolated.
Yeah. All right, so the first author is Priscilla Afonso. The last author is Clarissa Damaso, who I met in Brazil, and also buried in here. Other people I know Rolf Rennie. He's from your place, right, Rich?
Yep.
And I know, look at Rich Condit.
I'll be darned.
And Nisine Musace.
Yeah.
Did I say that right? Musace?
Nisine Musace.
Nisine Musace.
Close. Pretty good.
Why are we doing your paper?
Well, actually, I gave you a clue. Okay?
You sent it to me and I picked it, but I thought it was really cool.
Right.
So Rich didn't pick it himself. You probably wouldn't want
that would be self-serving, no.
Yeah. But if you did, we'd probably do it because it's cool.
But we get to ask you all about it and find out what you contributed.
I didn't contribute much. Okay, I was sort of I contributed money. So see all these guys with C's. Mm-hmm. That's Zhanhong Hu and Yaje Yang and Rolf and me and Nisine. That's all the part and parcel of the sequencing effort, because the sequencing was done in a facility that was set up by Rolf and run by these other two, Zhanhong Hu and Yaji Yang, and Nisine sort of directed that whole operation and did a lot of the assembly and annotation in collaboration with Clarissa. And then the other people, Patricia Silva and Layla Schnellrath and Desiree Jesus are part of Clarissa's operation, as is Marcia Attias. And Desiree, actually, Jesus, is actually now a postdoc in my lab. This all gets very convoluted, okay? I've got a Brazilian pipeline here through Nisine, and it's really nice because it's a nice collaboration that's held up over a long time.
It's in Rio de Janeiro, her lab, right?
That's right.
All right.
Clarissa was actually Nisine's PhD student, and basically hung around and continued on with his work in the same space as a professor in Brazil after Nisine retired and came and hung out with me.
Yeah, I had a drink one night with Clarissa, one of those lemon sugar drinks that Cotia, I forgot the name of it. It's on my iPhone. But she explained this whole pipeline. And it was very confusing, and I thought I thought I'd had too much to drink, but it was actually the story.
Well, it is confusing. You just got to play a little samba in the background, and it all makes sense.
All right, so why don't you tell us the whole story?
Okay.
Oh, the caipirinha. That's the name of the drink. Caipirinha. Okay. It's really cool. Go ahead. Tell us about this virus.
So, I think, as a sort of a general background, this is of interest because there are poxviruses circulating in Brazil. We've talked about this before, and we call it feral vaccinia. And the bottom line is that since we are no longer vaccinating against smallpox, people are no longer immune to it, and it creates a potential niche for the reemergence of poxvirus infections, and that has been happening in Brazil for the last 10 or 15 years. And there are vaccinia viruses that are running around, apparently. The most predominant one is a virus that has a signature deletion in the HA gene that Nisine and Clarissa actually were the first ones to characterize. And that virus, which goes by the name of Cantagallo virus, they showed is very similar, has the same, the vaccine strain that was being used in Brazil during the eradication campaign has that same signature deletion. And so the idea is that the vaccine probably established itself in the wild, maybe at a low level during the eradication campaign, and then reemerged when the immunity waned. But there have been reports of other viruses that are other vaccinia viruses, related, very closely related to the prototypic lab strain of vaccinia that's out there, WR, which is very curious because WR has never been seen anywhere but in labs. Okay, and one of the isolates that was characterized as vaccinia by one group was this, at least related to Cotia virus, but that was at odds with some previous findings going way back. So let's do Cotia virus now. This thing there's been a single isolate in 1961 from a sentinel mouse at a field station in Cotia, which is outside Sao Paulo, as we already discussed. And if you look up that original paper, it says, this is cool. It says that Cotia virus was first isolated from a suckling sentinel mouse of a family exposed for 48 hours at ground level in the forest. The family returned to the laboratory on March 3rd, 1961, and after 7 days of observation, one mouse became sick. Brain was harvested and a suspension was inoculated into other families of mice, and basically they were able to propagate it. And it, by complement fixation and some other immunologic technique, it didn't fit any existing arbovirus group, so they decided it was a novel virus and named it Cotia. They say later on that there were several other isolates. They say it's been isolated only from sentinel mice 40 times in 61, twice in 62, and 3 times in 63, and then it disappeared. But none of those other isolates, I don't think, were proven to be the same virus. So I think we can only count on this one virus.
So these mice are put in a cage out in the field? Is that how it works?
Right, sentinel mice. As a matter of fact, it sounds like a new litter is put on the floor in the field for 48 hours and then taken back to the laboratory and put under observation.
So how do you think they get infected?
Well, the idea would be that it would be probably insect-borne. And in fact, they say in this paper that they, or either this paper or the old paper or some others, that they checked a bunch of mosquitoes and didn't find any evidence of this virus. But don't know.
Yeah.
I mean, the most logical place is an insect bite of some sort, but don't really know.
So do you think the fact that it hasn't really been isolated subsequently from mice implies that mice might not really be the natural host?
My guess is that that's the case, that mice are not the natural host. It hadn't been isolated from anything. On the other hand, until this paper, really, we haven't known really exactly what to look for. With this paper, I think there's an opportunity, if you were really interested, to create appropriate primers or whatever to screen for this thing.
So the reason I asked is I went over this paper with my rotation student this morning, and she's from Venezuela, but she said, we looked it up on the map as to where Cotia is, and she said, well, that's an area where there could be deer and moose, or not moose, but, deer cervids. And on both of the trees that we're going to get to, the phylogenetic trees, that's kind of seems like what it might be close to.
Absolutely. So it could be an entirely different host. And the sentinel mouse infection could be entirely incidental. After all, it was newborn mice. And in fact, I think it doesn't affect adult mice inoculated by the intracerebral and interperitoneal routes showed no signs of illness, but did develop antibodies. So you're talking about effectively an immunocompromised animal, the newborn mice. So it's probably I would doubt if it's the natural host, rodents, but you don't know.
Right.
So at any rate, in subsequent years, over a long period of time, several other groups had a crack at characterizing this. And the characterizations used whatever techniques were available today at the time. And initially, this involved serum cross-reactivity. And basically, although there was a little bit of cross-reactivity with some groups, it certainly didn't seem like it was an orthopoxvirus, which would include vaccinia. But it also looked like it was fairly unique. Other characterizations included restriction enzyme mapping, and it also looked unique by that criteria, and some very limited sequencing of a couple of genes, and it looked unique by that criteria. So several different groups looking at it thought that it looked like maybe a unique virus. And then comes this report from this one group that says, no, it looks like vaccinia virus. And so there's been some suspicion, actually, that's even been discussed in the literature that maybe this sample contained two viruses or something like that. And Clarissa and her colleagues decided that they really wanted to sort this out. And so they got some of the original passaged material from, I forget exactly the source, it may be the Institute of Oswald Cruz. Kindly provided, from the Institute Adolfo Lutz in Sao Paulo, Brazil. And they basically did two things. They did a biological characterization of the virus, and then they sequenced the whole thing. And interestingly, what they got out was a virus that is, in fact, different phylogenetically, different enough from any other pox viruses that has since been, that has previously been reported to put it forth as a candidate for a unique genus of pox viruses. And they also found that the biology is a little bit different, I suppose. Well, it's different than vaccinia anyway, in a fashion that makes sense relative to what's been published. In particular, although if you look at yields of virus in a time course of infection, or if you look at protein synthesis or DNA replication, it seems to follow pretty normal kinetics. But if you look at what happens to the cells, it's very slow to create a cytopathic effect, and it makes only very small plaques after an extended period of time. And they speculate on why this might be based on the genome sequencing, and it's possible that there's one gene in particular called B5 that's important in spread of the virus that's missing from this virus. And so they're thinking that the mechanism of spread might be slightly different than vaccinia, and that would account for the difference in cytopathic effect and for the small plaques. But importantly, what that means is that if you ever had an opportunity to have vaccinia and this thing together in the same culture, and you were trying to isolate virus out of it, it would be very easy to selectively isolate the vaccinia, because that's going to make the big plaques. All right. Mm-hmm.
So there are 9 genera currently, right?
Yeah. So you're looking at the phylogenetic tree.
And this would be a new one. And your guy, vaccinia, is an orthopox, right?
Right. So the orthopox viruses include cowpox, variola, monkeypox, and vaccinia virus. So variola is smallpox. Monkeypox is very closely related to it. And cowpox and vaccinia are, of course, related to those. These original phylogenies, or the original groupings, were based to a large extent on serology, but they're borne out by the genome sequencing. And the orthopox viruses are a very tightly grouped bunch of viruses, and a significant difference from what they call the, let's get it right here. They talk about a clade, and I can never remember the...
Oh, it's the CSYLC.
Right, CSYLC, which is...
the genera.
Capripox, Suipox, Yatapox, Leporipox, and Cervidpox. That's these 1, 2, 3, 4, 5 different genera that group together on a large phylogenetic tree. And they call these a clade and say that Cotia virus groups, and it does, groups along with that clade. Now, as you pointed out, Kathy, that includes capripox are like cloven-hooved animals, goats, sheep.
Goats, yeah.
Suipox is sheep. Cervidpox, that's like deer. They're hooved animals with antlers. Leporipox is rabbits. And Yatapox shows up in monkeys. So there's the species distribution is pretty wide. And each of these genera are distinct from each other. You can see that. And Cotia virus kind of fits in the middle of this and is distinct from any of them, and is a significant difference from the orthopoxviruses or the parapoxviruses, which we're going to say a little bit about later, or a couple of others. You'll notice down here, way off in the wings, are the foulpoxviruses and molluscum contagiosum. That's a human virus, MOCV, and crocodile pox, which is way out in left field. They're calling that unclassified as well.
That's CRV, huh?
Yeah, CRV.
So this is just entirely by sequence.
That's entirely by sequence. And in fact, it's, both of those trees are by sequence. And in fact, I was thinking about this. The way they did the sequence, so the way the poxvirus genome is structured, there's a conserved block of genes, and that's usually conserved in the central region of this linear genome. And we're talking about 200 genes total on average in these viruses. And the conserved block, if you don't include the outliers like foulpox and molluscum, the conserved block is like 90 genes. If you include the outliers, the conserved block is more like 40 genes. And they took the 90 genes and made a catenated, strung them all together and used that as their, for all of these viruses, as their probe to look for the relationships. Now, the genomes also, the poxvirus genomes also contain a bunch of what we call variable genes that are usually housed on the flanks of the genome. And those are the conserved genes are the nuts and bolts of pox replication, transcription enzymes, replication enzymes, et cetera. The guys on the flanks are responsible primarily for characteristics of host range and pathogenicity and that kind of stuff. And the comment that they make in this is that these trees are constructed on the conserved genes, and by those criteria, this thing clusters with this CSYLC clade. But interestingly, the genes involved in the variable genes are more similar to the orthopoxvirus genes. So it's kind of, in a way, a mix of the two. It would be interesting, actually, to see a tree with all of the variable genes catenated, but I don't know how you would do that with all of the viruses, because the variable genes are so variable, it would be hard to make a catenated structure with them. At any rate, it's almost as if it's either ancestral to both the orthopoxviruses and this clade, or maybe a recombinant way back in ancient times between the two, because it has some characteristics of both, though with respect to the conserved genes, it clusters with these CSYLC clade.
So now that you have this sequence, then other people, when they discover viruses, can add to it.
Right.
Now that it's there, we should mention that it's 185,139 bases long. So it's not the biggest pox virus known, right?
No, that is not the biggest known. Some of the foulpox viruses get up to 300 kilobases.
Are there any? Did they find any sequences of unknown Because I noticed there's a big table
Yes.
of annotated genes and a
Right.
lot of the functions there.
There are some Cotia virus genes that have not been found in other poxviruses and have not been found elsewhere in the database. So they probably provide some of the unique properties of this virus, or could provide some unique properties of this virus. You can't really do this, but it would be interesting if we knew enough so that you could look at a lot of the variable genes are immunomodulators.
Yeah.
All right, that the virus uses to tweak the host, and in some of the viruses, some of the immunomodulators are very species specific.
Sure.
It would be interesting if you could know enough, just based on the sequence, to look at some of these variable genes and predict what the host would be based on the immunomodulators. Okay, but we don't know enough to be able to do that.
It makes sense that the immunomodulators would vary according to
Right.
the host, right?
Right.
I think they mentioned two of them, and they seem to be, similar to ferret and mouse, CC chemokine-like proteins.
Right. Okay. Right.
Ferret?
So-
Did you say ferret?
Yes.
Yeah. That gets Vince's attention.
Yes.
It's already published.
I had a comment because I was struck by the fact that all this conserved block of genes is in the center of the genome, and that's kind of true for adenoviruses as well. So the DNA replication and the capsid protein genes and all that are in the center, and then the early region genes that tend to be those that have to interact with the host are out at the edges. And I wonder if that's a common pattern or just kind of convergent evolution.
No, I think so. I can't remember exactly, but that theme came up in another virus as well.
Yeah, it did.
So my guess is that that's, and I wonder if that has something to do with recombination.
Mm-hmm.
I can't quite think this through, but somehow it makes sense to me that if you cluster all the conserved stuff in the middle and leave the non-conserved, the variable stuff on the outside, that that makes sense in terms of recombination, that it would be easier to conserve the stuff in the middle and swap the stuff on the outside.
Mm-hmm.
It seems to make intuitive sense to me. I'd have to draw it out to make it make more. I'll talk to my recombination buddy, Dave Evans, and see what he has to say about that. But at any rate, I have seen this before in some other viruses.
This came up in Mimi virus.
Is that it?
We did a paper where when they pass it in culture, it loses a lot of its genome, if you remember.
Right, okay.
Honey, I Shrunk the Virus, that was the name of it. And then it was from the ends, and then someone wrote in subsequently saying that you see the same thing in adenovirus.
Okay.
Okay.
Yep.
So I had one technical question, Rich. They propagate this at 34 degrees C. Do you know why?
Just because it works best. And as a matter of fact, there have been times when we've had, and my guess is that this, well, I suppose it could have something to do with the natural biology of the virus, but we've even seen this, or its host, yes. We've even seen sometimes when we've had, and I think Nassim has imported this into our lab based on his experience with this. Sometimes when we've had trouble growing various variants of poxvirus or various mutants in our lab, we'll drop the temperature down, and sometimes that helps. So it could be just a laboratory, I don't want to call it an artifact, but it could just be a laboratory phenomenon, or it could be relevant to whatever the host actually is. I don't know.
Do you have this virus in your lab now?
I'd have to ask Nassim. I don't think so.
I presume that
we've had the DNA.
Clarissa's group will continue to work on this, right?
Yeah, they even mentioned that they're interested in some of the unique features of the biology, in particular the spread, and whether the spread mechanisms of this virus, because it seems to have this difference in cytopathic effect, whether the spread mechanisms are the same or different than other poxviruses.
Mm-hmm. Neat.
So, new virus.
New virus, a lot of data in this paper.
Yeah, and
Wow.
well, it's been going on for years. It's been
a lot of nice kind of classical virology too.
Yeah.
Yeah, I thought the same thing.
I like that.
Things you usually don't see anymore.
Right.
Growth curves, protein gels, plaque assays.
Right.
We have a new faculty member here, and he works on influenza. and he hates plaque assays, Rich.
Oh,
He wants to develop, like fluorescent assays to measure.
oh come on,
He said he
plaques.
hates it. I don't get it.
Yeah.
It wasn't trained properly.
No. Well, twist him around.
I will. I'm working on him. All right, that's great. I would be proud to be on this paper.
Yeah, it was. I'm pleased that this came out the way it did. I'm proud to be a part of it, even though I didn't. Mostly what I contributed was the money for the sequencing. But I had a lot of discussions and read a lot of drafts and that kind of stuff. It's been a pleasure to be part of.
It's nice. Good job. Well, I also I picked another pox paper. It's something I think you had put on the agenda.
Me?
I think so. Didn't you put
I don't think so. No,
Maybe
I
it was
didn't.
Alan?
This was a surprise to me. I hadn't seen this before.
All right. Well, then if it's not you, I'll take it off.
Oh, no. This is really cool.
This is cool. This is actually, I guess it must have been Alan then, because only the three of us add to this one document. It's from the Morbidity and Mortality Weekly Report. It's titled Human ORF Virus Infection from Household Exposures in the U.S. from 2009 to 2011. I figured you'd like this, Rich.
Yeah, this is great.
So I'm going to let you take it.
Oh, I get to take it? Okay.
If you don't want to. It's
No, that's cool. So this is, as it says, ORF virus infection from household exposures
what's
over
an
a
ORF
2-year
virus?
period. Okay, so...
Does that mean open reading frame?
No. As a matter of fact, I don't know why it's called ORF.
I
Somebody
do.
will write. You do?
I do. I
Why?
looked it up, and you go back far enough. I found this paper. I put it in the show notes, and it's the Anglo-Saxon word for cattle. ORF.
Okay.
Wow.
So this is another poxvirus, and in fact,
Which genus is it?
I closed it down. Where is that? It's a parapoxvirus. Have you still got that? I got to go back to that.
The earliest mention of this was from 1787, this ORF disease, according to this paper.
Okay.
Not the one that we read, but the history paper that I put in the show notes.
I guess the lesions are quite characteristic, right?
Right.
Yeah, so if you look on that phylogenetic tree from the last paper, you can see parapoxvirus down near the bottom.
Mm-hmm.
And it's got ORFV, that's ORF virus. And then BPSV, I think that's bovine pustular stomatitis virus or something like that. So you can see that phylogenetically, they're kind of out in left field as well.
Yes.
And so they're pretty different than a lot of the other pox viruses that we think of. These are viruses that show up mostly in ruminants, cattle, and sheep. And their disease is limited primarily to just these single cutaneous lesions. They're, I think, transmitted primarily by direct contact into open breaks in the skin, and they cause these pustular lesions that look like pox that we're familiar with in something like smallpox or in a smallpox vaccination, except they do get mighty big and ugly. And actually, there are other stray parapox viruses around. As a matter of fact, I co-mentored a student some years ago who isolated and studied a parapox virus from seals that was showing up in some seal rehabilitation facilities out in California that they were trying to deal with. At any rate, this MMWR paper reports on 4 different exposures, cases of ORF infection in the United States, and what they all involve is people who, for one reason or another, are slaughtering or butchering animals on their own property or in their own homes. There's one woman who slaughtered a goat near her home in Greece, but then made a trip to Pennsylvania to visit her son. And the lesion that she acquired during the slaughter was, they reported it while they were there and got it taken care of. Another guy in Massachusetts was assisted in a lamb sacrifice for a Muslim holiday, and he held the lamb's head in his left hand and developed a lesion on that hand. There's another man of Ethiopian descent in Massachusetts cut his thumb with a knife while slaughtering a lamb as part of Easter festivities. And the last one was a pregnant woman from Sudan who cut her right hand on a bone while preparing a lamb's head at her home in Virginia. So all of these are, I would call, cultural exposures, where people are, for one reason or another, slaughtering ruminants or butchering ruminants in their homes and get some of this virus in a lesion. Now, the animals that they slaughtered didn't necessarily have visible signs of disease, but I think it's not an uncommon infection in these animals. And I don't know how easy it is to detect disease in the animal, but they picked it up. And these pictures are pretty startling.
Yeah, big lesions.
Okay, big lesions. Wow. Big pustular lesions. But
it says they're not painful in one of these cases.
Yeah.
It's kind of hard. No fever, no pain or tenderness.
Right.
Wow.
A different one does say it was painful.
Another one did, yeah.
Yeah.
Right.
Yeah. So depends probably where it is.
And this is it's an occupational hazard of butchers, the ORF lesions. And interestingly, you can get reinfected. The immunity is not long-lasting. And there's even some suspicion in the case of the sealpox and some other diseases that there might even be a persistent infection in the animals. And there's recrudescence of the infection when the animals become immune compromised, though I don't think there's any evidence of that in humans. In humans, basically, once you know what it is, in most cases, you just watch it, and it's self-limiting. It heals and goes away, and that's fine. And that was the case with all of these. One of the things that I was impressed with is that well, of course, they wouldn't have made MMWR, I suppose, otherwise. But in all of these cases, there was some sharp physician on top of this who said, aha, I'll bet you that's ORF, and sent off material to the CDC to be characterized by a PCR reaction for confirmation that it was ORF virus. And in a couple of these cases, the description sounds like the diagnosis was made fairly quickly, and they figured it out, clinically fairly quickly. And what they point out in the editor's note is that it's important for physicians to be aware of this, so that they don't, for example, treat unnecessarily for antibiotics or cause undue psychological stress by telling a patient it's something that's, not self-limiting or nastier than ORF virus. So it's important for people to be aware of this.
One of these patients got an antibiotic, though, mupirocin.
Yeah, I think that's not necessarily a bad thing in some cases, because these might be prone to secondary infections, and so some of that antibiotic treatment could be prophylactic for secondary infections. They noticed that on the analysis of some of these lesions that at least some of them had a little bacteria associated with them as well. And in one case, they actually surgically treated this thing, which I think if they get really nasty, that's not an inappropriate thing to do.
Yeah. Would you give these individuals an antiviral?
Well, now that's an interesting question.
Now we have some, right? Now
we have some. There's cidofovir, and there's ST246. Cidofovir, the licensed form of that has to be taken intravenously. This is not a life-threatening infection. So unless it went nutso in somebody that was immunocompromised, I don't think that that's indicated. And actually, now that I'm thinking of it, I don't even know if ST246 works on ORF virus. I would imagine that they've tested it, but I just don't know the answer.
So that would be the case. If you had an immunocompromised person and this thing went crazy, you'd have to...
Went crazy, yeah. Right.
They also say it's bad if it gets near the eyes, right? Is that? Yeah,
you could get an infection in your eyes, and that's uncomfortable. It can cause some permanent damage, but usually those infections heal up okay. So,
Well, so everyone, if you are slaughtering goats and you get a lesion sometime later, this is what it is.
What the other editorial comment they make is if you're slaughtering goats, you might want to consider wearing gloves.
Yeah.
Right? And, my guess is that what's key in all of this is if you're a physician, is the patient history.
Yeah, exactly.
Okay, because what's common in all of these and unusual is the slaughtering and butchering of animals outside of a normal facility, the people doing it themselves. So that would come up as a flag for something like that. But the physicians have to be aware of it.
Many years ago, I was at an Italian dinner, a big banquet, and at one point, there were a lot of people there. They brought out a tray, and they uncovered it, and on this tray was stacked about 50 lamb skulls.
Oh, great.
With I guess, the brain in it, and then everyone ran up to get one.
You ran the other way.
Yeah, I didn't move. I said, this is terrible, but this is apparently a delicacy. I don't remember what time of year it was, but these were cooked. But whoever cooked them might have been at risk for ORF. Mm-hmm. Interesting. Okay, that's a cool story.
Yeah, I love the MMWR stuff. It's great stuff.
We'll put a link to it so everyone can see these nice pictures here.
Yeah.
All right, our final story is one I picked up because this seems to be prion week here, or prion, as I gave a lecture on Monday, yes, to my virology course on unusual infectious agents, and one of them is prions.
So, Vincent, I have a comment about pronunciation because I have never known. And so I have 4 dictionaries at my disposal. And 2 of them list both pronunciations, prion and prion. And each of those dictionaries lists one or the other as the first pronunciation as correct. So that doesn't really decide things. But 2 of the dictionaries only list one, and they both use prion. And it turns out that prion is also a bird. So if you really want to distinguish between these protein particles and birds, you should probably say prions. But that's not really definitive.
What I told my class is all these words are made up, so you can pronounce them any way you want.
Right.
Unless you ask the person who made up the word. In this case, I guess, was Stanley Prusiner. You could ask him, but I think it's like all the drug names that are made up. What's the difference how you pronounce them?
Right.
It's fake. It's not like it's a real word. So on Monday, I gave a lecture. Then the next day, one of my students emailed me a story saying, which said that a cow had been diagnosed with prion disease out in California, mad cow disease. And so then that was the day I was looking for stories for TWiV, and I came across a prion article, also in the Journal of Virology. And so I picked it because I thought it would be interesting. And this is called Protective Role of Interferon Regulatory Factor 3 Mediated Signaling Against Prion Infection. And the reason I picked it is because, well, let's talk a little bit about prions. I think we have had a TWiV on prions, but it was a long time ago. Prions are proteins that can change shape. They are shape shifters. They're zombie proteins. And in mammals, they can cause disease. They're pathogenic. So
these are proteins that we ordinarily make. They're proteins that we use every day.
We have normal versions of these proteins, all of us, and they're important in us.
Right.
It's worth mentioning, though, that yeasts and fungi also have prions, and those are proteins that change shape, and they don't cause disease, but they're very beneficial for the organisms. And these, for example, will change shape in response to environmental conditions, and they allow the yeast to do things that they couldn't otherwise, like use a different carbon source to make energy or something like that. So they're really cool in those organisms. But the first ones discovered were mammalian prions, and that's because they cause these transmissible spongiform encephalopathies, TSEs. They're totally fatal neurological degenerative diseases like Kuro and Scrapie in sheep, Creutzfeldt-Jakob, and they're caused by misfolding of this Scrapie protein, this prion protein. So we have a normal form, and the pathogenic form is a conformational variant of the normal protein. And so you can get infected with these proteins, and then what they do is they get into your brain and change your normal prion protein into an abnormal one, and eventually you get the neurological disease. And spongiform comes from the appearance of the brain. It has all these holes in it. So we have this normal form, and sometimes so in Creutzfeldt-Jakob you can acquire the pathogenic form by say a corneal transplant or a transfusion of some kind from someone with the prion who didn't know they had disease. In England, there was an outbreak of variant Creutzfeldt-Jakob from people who ate beef that had been taken from cows with bovine spongiform encephalopathy or mad cow disease. There are also familial forms where you inherit a mutation in the gene, and eventually that causes the protein to misfold, and then you develop the TSE. And then there's sporadic forms where you just develop it, and there's no mutation, there's no association with getting in the protein by some way. And that's, in fact, what the cow in California apparently had, a sporadic form of BSE.
So the outbreak of BSE in people in England,
right?
Are we convinced that was actually BSE? And because there's a certain level of spontaneous prion disease in people, and these things are difficult to distinguish one from another.
Yeah.
And yet I gather that they had criteria by which people felt they could reliably distinguish between the bovine origin disease and spontaneous human disease. But I'm wondering how convinced we are that there was really an outbreak of bovine disease in England. I guess people are reasonably convinced of that.
It's pretty good. So there's a very nice epidemiological correlation. There's a long incubation period, but you could see a spike.
A blip.
Right after.
It wasn't
the cows.
very big, was it?
No, it was less than 200 cases, I believe.
Okay.
The disease is different from normal Creutzfeldt-Jakob. It has a different incubation period. That's why it's called variant Creutzfeldt-Jakob. So there are enough indicators which say that it was above the basal level of CJ, I think, and had specific features. And of course, arose the cows had BSE because they were fed ground up carcasses from other animals who presumably had a TSE of some sort.
Right. So when they figured this out, they changed the feed.
Yeah, they stopped doing this. It's called mechanically disrupted protein. They would break up the spinal cord with high pressure water, and that would release all the prions. And they stopped doing that. And
actually, my understanding is that the reason that they've done that for a long period of time. However, prior to the outbreak, they used to render that stuff, that is, heat it up to a certain temperature to make tallow. But then the market for tallow went away, so they no longer did that. So the rendering process, which ordinarily would, to some extent at least, inactivate the prions, went away. So the same feed actually now was pathogenic in the cattle.
Right.
Then they had to change the feed altogether to
they did.
to make it go away. Which actually I would call this sort of an epidemiologic success story, right?
Yeah.
Figuring this out, saying we got to take action.
As soon as they changed the feed, the outbreak in cows went down. What's amazing is that the reason they give the cows meat is because they grow faster, right? You can give them grass and corn, I guess, or just grass. And in fact, grass-fed beef here is a delicacy, right? And it tastes great, apparently. But the farmers want them to grow quicker, so they give them protein. That's not what cows usually eat out in the fields. And once again, we mess with things. Anyway, I'm diverging. So there was a case out in California. I had taught this lecture on Monday, and one of the things I told them is that there's no immune response to prions. They're your own protein. They're recognized as self. And in fact, I had just read Stan Prusner's revised Fields chapter on prions, and it just says there's no immune response, no cellular response, there's no antibody response. But I find this paper which says there may be an innate immune response to prions, so I thought this would be cool to just go review. And what they say here is that there is There's no antibody response. There's no cellular immune response. But there are inflammations going on in the brains of animals with these TSEs. Mm-hmm. In particular, they say gliosis is a major characteristic of TSE. It's a kind of inflammatory response. So they say, hey, there must be some kind of immunity. Gliosis is proliferation of astrocytes. And so that's what their basis is here. And there's some evidence previously that there may be innate immune signaling. Now, everyone remembers that when viruses infect cells or bacteria are around, they stimulate innate immune responses. There are receptors that can detect these foreign molecules that viruses and bacteria carry. There are toll-like receptors. There are cytoplasmic helicases. And we've talked about these on and off in the past. And the way these things work is these sensors, like toll-like receptors, will sense either an RNA or a protein or a lipid as foreign, and they start a signaling pathway. And eventually, you activate transcription factors that go in the nucleus, and then they make interferon. And the interferons then induce an antiviral state. So that's what this paper is about. They knock out one of the genes encoding one of these transcription factors called IRF3. So this is a protein downstream of the sensors of the innate system. And they have mice that lack it. And they know.
So IRF3 is kind of in the middle of this pathway. It's downstream from the sensors, but downstream from IRF3 is going to be transcriptional regulation of a whole array of genes that have to do with the interferon response, right?
That's right. And, leading to IRF3 is a whole bunch of phosphorylation cascades mediated by protein kinases. And IRF3 eventually gets phosphorylated itself. It's normally in the cytoplasm. And when it's phosphorylated, it makes a dimer. It goes in the nucleus and then turns on mRNA synthesis of various sorts. So in this paper, they knock out the gene in mice for IRF3. These mice have been around for a long time, and they infect them with prions. So you can infect mice intraperitoneally with prions, and they will develop a TSE. And so they do a variety of things. They look at the survival time, because these prions will kill the mice eventually. So they have 3 different strains of prions, including the cow strain, BSE. And for example, if you infect these mice with wild-type mice with these prions, they die in like 280 days, 380 days, 321 days.
I do not want to do these experiments.
This is ridiculous.
These are expensive, long experiments.
It's over a year.
Yeah.
It's crazy. Anyway, if you take an IRF3 knockout, it takes a little less time for them to die.
This is a really crazy thing. In particular, if you look at the table, and you see that the survival period with one prion in wild-type mice is 281 days, plus or minus 15 days.
Yes.
All right? And that's 9 mice. And in the IRF3 double knockout, it's 257 days. So that's, 24 days different? Plus or minus 8 days, right? Now, they do all the statistics and stuff, but those are the kind of data that you're dealing with. And this is how they speciate these prions, too, with symptoms of the disease that they cause and how long it takes to kill them. My goodness. I don't know if I could deal with this.
Now, these are tough experiments.
That's how the experiments are done.
Yeah. And a number of labs have done these, and they take a long time, and you have to give it to them.
That's right.
I mean, Stan Prusner has a huge lab doing this, and it's extremely expensive.
Right.
Every time his grant comes up for renewal, people are like, oh my gosh, how can we spend this much money? But if you want the answer, you have to do it.
And prion disease is important, okay? It's the whole prion deal and amyloid deposition is going to be important for much more than just prion disease. So this is an important study. The graph of the survival is much more dramatic than the table. Yeah. Okay, that's easier to swallow.
Yeah. Mm-hmm. So then they also look at the amount of the protein in brains. So you can distinguish between your normal prion protein and the pathogenic form, because the normal form is totally digested by proteinase K, whereas the pathogenic protein is resistant. Only the N-terminus gets clipped off, actually. And they don't see any differences in the amount of the pathogenic protein in the wild-type mice or the knockout mice. They do a lot of histology of brain sections, and they find in some cases, depending on what they're looking at, you can look at certain cell types like astrocytes and microglia in the brain. Sometimes they find differences, sometimes they don't, and the differences are rather subtle. So, for example, I'm looking at figure 2. There's a slight difference in GFAP, which is looking at astrocytes. You can see there's more astrocytes coming up sooner in the knockout mice than in the wild-type mice.
Right.
In other cases, also the vacuolization, the spongy appearance, is a little coming on earlier in the knockout mice.
In general, most of the differences that they describe are in terms of the kinetics of the infection.
Yeah, that's right.
Yeah.
There's no really gross difference. It's
No.
the kinetics that are different.
No, in the end, the mice all die.
Right.
And they have TSE. So this is not like it's doing anything to alter that, but you get subtle differences.
This I was impressed with. I mean, this is normal prion stuff, but I don't ordinarily look at this stuff. I was impressed with this figure 1B where they show do Western blots with antibody to the cellular prion protein and the pathogenic prion protein, or they use antibody to the prion protein plus and minus proteinase K, so you can see how this thing is protease resistant, and it's really dramatic.
Yeah, that was an important finding years ago that helped them distinguish the two. Yeah.
Right. You can also see that when looking at this protein, it's a very heterogeneous. It runs really funny on gels. It's not a very homogeneous species.
Right. Multiple bands. Yeah.
Yeah. It's just a real pain to work with.
Then they do a number of experiments in cell culture. So it turns out with certain cells, you can put these prions in them. What you do is you take the brain extract of an animal with a TSE, and you just put this extract on the cells, and the cells will take up the pathogenic prions, and they will multiply inside the cells. Now, you have to overexpress the normal prion protein in these cells in order for this to happen. But when you do that, and they use a neuroblastoma cell line, you can put in a brain extract, and you will get amplification of the pathogenic prion in cells. In fact, we did a TWiV a while ago. It was called Darwin Gets Weird, I think.
Right.
Showing evolution of these proteins in cells. Mm-hmm. In the absence of a genome. It's really cool. Anyway, if they overexpress IRF3 in these cells, it decreases the amount of the pathogenic protein that you find. So it decreases its multiplication. And if you remove IRF3 with siRNA, you get more of the pathogenic protein.
Right. So it all makes sense.
So it makes sense in the sense that somehow IRF3 is playing some kind of role. Of course, what it's doing, nobody knows. And that's what these guys are going to work on in the next 100 years, probably.
Right. And since IRF3, regulates a whole bunch of genes, it's going to be a complex problem.
So the last thing I just want to point out is that they've shown before, others have shown that interferon doesn't make any difference in the propagation of prions. So we know that IRF3 is a player in the induction of interferon, but it also is in the pathway of inducing non-interferon, innate genes. It's various other cytokines. So probably those are the ones that are playing some role. And it'll be important to figure that out because maybe you can translate it into a therapy. Because at the moment, when you develop this kind of disease, a TSE, by the time you have symptoms, you're gone. You're going to die. They're uniformly fatal. So we either have to pick it up earlier or figure out how to treat it.
And somewhere upstream from IRF3, and what was it, TLR4 or something like that? Mm-hmm. There is some sort of receptor that senses this as not normal.
Right.
And it's not known exactly how that happens, but at least you know it's not normal.
We don't know what senses the prion. That's true. And so I think it's going to be the next step to find out which of these sensors, which of the toll-like receptors, which of the cytoplasmic helicases maybe, which of those are involved in the sensing of it. So it's a cool story.
I had one comment that I wanted to make for people who are doing knockdowns, that in Figure 5, they say that they knock down the IRF3 significantly. But if you look at the graph or the gel itself, the blot itself, it's not particularly convincing. But yet they do see an effect on the ratio of the prion protein, the mutant prion protein, to the wild type. So even a little bit of the knockdown, what I would call a little bit of the knockdown, did give them the effect in the direction that they expected based on their hypothesis. Right. And the other thing is that I knew that the American Red Cross used to screen donors if you had spent too much time in England. And it looks like now, and I haven't given blood in a while because I used to get hassled about it and was only from visiting there for meetings. But now it's a cumulative time of 3 months or more from January 1980 through December 1996. You would still not be on their good blood donor list.
Yeah.
So
that's why they freak out when there's a cow that gets diagnosed with BSE anywhere now, because they don't want it to go. And this cow into the food chain. This cow in California was a dairy cow. So they said, don't worry about it, because you don't eat the meat and the.
Right, and there was something about it being not the normal BSE prion, or it was a variant of some sort.
Yeah, it's called atypical BSE. It's called by a variant which you pick up by a proteinase K digestion, basically, and it's a spontaneous kind, so it's not induced by contaminated feed.
Right. So this was not a sentinel cow.
Yeah.
Right.
That's right. There's a cow who died. It was a dairy cow that died, and they randomly select cows, when they die for testing, and they tested this one and turned out to be positive.
So, and Vincent, when you were talking about your Italian dish with the lamb brains, this is what I thought of. Not the ORFvirus, but some kind of scrapie or something. I wouldn't.
Well, that's interesting because there was all this fuss about the bovine spongiform encephalopathy getting into people, and yet scrapie has been around in sheep forever, and as far as I know, there's no evidence that jumps to humans. Right?
Yeah. I was just thinking the same. There's no evidence. There are some big species barriers among these, and the cow happens to be pretty loose in that sense, which is why we get it from cows. And you can take the cow prion and put it in mice, like what they did in this paper. But other cross-species infection is not as good. So, I don't know of any from sheep to people.
The thing that boggles my mind is that, so this thing, if you get the cow prion, that triggers a conformational change in your own prion.
That's right.
Okay, which is propagated now, and the cow thing is gone. Right? You're just propagating that conformational change. And yet that change can be distinct from a different sort of conformational change that you might get in Creutzfeldt-Jakob disease or something like that. Right?
Yes.
As if the species, there's a species-specific conformational change that is propagated.
Yeah, it's amazing.
That's hard to get my head around.
Yeah, it's incredible. This is an amazing phenomenon. That's why I love giving that lecture. It's so cool. But things progress slowly in this field.
Yeah.
It takes a long time. So that's cool. That's why I thought this would be neat. It's a good week to do a prion paper. And I didn't know that innate immunity was involved, so I really learned something.
Yeah, that's good to know.
It's cool. Yep. All right. Let's do a few email here. I'm going to let Rich take the first one.
Okay. Andrew writes, good, insert appropriate distinction regarding the time of day you receive this, gentleman of TWiV. I see. That could be morning, afternoon, evening, whatever. It's afternoon. Good afternoon, gentlemen of TWiV. I've been an off and on listener to TWiV for what must have been before 2009, in the middle of my undergraduate years. This podcast serves as an example of the power of the medium to deliver focused, higher-level discussion on topics too complex to survive on traditional broadcast media, but still casual enough for the non-scientists. So thank you for your excellent service to us, the listener. The question, concern, comment I'm writing about is this, in capital letters. Andrew says, graduate school is scary. I graduated from a local state school with a BS in biology in 2010 and have been out of school for just under 2 years. I've maintained some, probably about 5 to 10 hours a week, of activity in the population genetics lab I worked in prior to my graduation. Some family issues kept me tied to the area and reluctant to apply to graduate school, but recently those issues have resolved themselves, and I now find myself looking to take the next step. This is very frightening, and I find it difficult to proceed. How do I find what master's or preferably PhD program is right for me? Am I doomed? What do I do if the program best for me doesn't think I'm best for it? It seems too late in the year to apply for anything. Most programs have closed applications by now, and those that haven't have disclaimers stating that funding is usually assigned already. Should I bide my time and wait? In the meantime, I'm currently applying to the IRTA post-baccalaureate training program, which seems like a great program to get into in order to help bridge the gap between undergrad and PhD, and may even deserve a mention on the show. And he gives a link. Any advice on how to proceed when caught in this quagmire would be greatly appreciated and would likely help any listener also caught in the same post-undergraduate, pre-graduate morass. Thank you. Whether you read this or not, your podcast has been a huge influence on me. Please never stop. P.S. I actually forgot to include the field of study I'm interested in because I meant to move it around and then forgot to re-include it. My long-standing interest in genetics managed to fuse and include virology, and so ideally I'm most interested in virology, hence looking for TWiV's perspective, with a focus on evolution and genetics. There you go.
So graduate school shouldn't be scary. And he's right. There is something of a season for applying to graduate schools. For instance, our deadline here is December 1st for the following September for doctoral programs. Our deadline for master's program is later, but it is also already passed for starting this September.
You have a master's program?
We do have a master's program. We're just in our second year of it. That's
right. We discussed that.
Right, but it's a program where the students are paying their own way. So
right.
It's definitely something for somebody who knows this is what they want to do, and sometimes it's because they want to get a little more training that might make them more marketable for either graduate school or medical school, or something like that.
Right.
So.
Okay. Sorry, I interrupted you.
Well, that's all right. So is he doomed? No, he's not doomed. But yeah, it may be too late for this year. So this NIH-based post-baccalaureate training program is a really good program. We get a number of applicants to our umbrella graduate program every year that have done post-bacs either at NIH or at places that have post-bac training grants, of which Michigan is also one of those. So that's a good way to get some lab experience. But you could also just try and get a job working in someone's lab. Funding times are tight, so they might not have a paying job, but you might even be able to do some volunteering and do, whatever kinds of research undergraduates do, but you're already, post that. But so that's a way to.
He seems to be doing that, right? He's working in a genetics lab.
That's right.
Yeah.
He did say, 5 to 10 hours a week, right?
Yeah. So it's too. Yeah. It's you're right. It's too late now, but that's okay. It gives him time to look around and find what he wants to do. How you identify a program? I don't know. You talk to people, you cruise around on the internet, and look for programs, and you don't worry about whether or not they're going to take you. If you got a halfway decent record, you can probably find a program and just go ahead and apply. It's a jungle out there. Some people are going to turn you down. Some people are going to accept you.
Right.
But if you don't apply, what is it? Joe Sambrook told me years ago. If you don't ask for it, they're not going to give it to you.
Right.
He was talking about grants at the time. Okay. So.
I think if you're interested in virology, just go to a program where you can work in a virology lab. You go to a microbiology department that has a lot of different interests, and make sure they have virologists there. And you might find that once you go, you'll do something else that catches your interest, but that's fine. But it sounds to me like you want to do virology, so just apply to a program and do it.
Apply to a bunch of programs,
yes.
And there will be an interview process, and go check them out. Okay, because this is what happens. You apply to a bunch of programs, and nowadays, in order to compete for graduate students in this field, the various programs really have to interview people, and they have to actually fly people in for the interview. So you won't have to pay for that. And if you apply for a bunch of places, you'll get interviews, and that gives you an opportunity to go and talk to the faculty and talk to the students and look the place over, and you'll learn a lot. And anything that looks interesting. Go ahead and apply. Apply to a bunch of different places.
Yeah, find places that have research that you're interested in. And also you have to consider some places are more competitive than others, and you have to apply to ones that are commensurate with your record, although you should shoot for some high ones and take risks for sure. Mm-hmm. Just like college, you have to take some risks, and you have to have some sure ones and some in between. And just do it. Because if you want to do virology, that's the way to do it. Just do it. And then once you get to a graduate school, that's another story entirely. And I would send you to Alan Dove's post on this on his blog called Some Advice for Stressed Out Graduate Students.
Oh, I haven't seen that.
Look at that, because it turned out that Alan Dove was very stressed out in graduate school, and of course he was my PhD student, and I had no clue, which often happens. And it's good that he worked it out. But just get in first. I don't want to scare you, Andrew.
I was very stressed out, too. But that had more to do with me than graduate school. That's just a matter of figuring out how to live.
Yeah, it's a hard thing. Ellen has a good post on it. Check that out. The next one. Can you read the next one, Kathy, for us, please?
Sure. Jim writes, Hi, Vince. Last November, you had Science 360 Radio as a TWiV pick. There are now over 100 program links there. That provides a concentration of material you can point folks toward to more easily find out what they want. I hear many podcasts from these places that are good, but some sites always stand out, like TWiP, TWiV, TWIM. Others periodically have great shows or a segment of a digest, and some have a huge number of past programs. So if you have an interested listener, I don't know where you send them, especially if their time is limited, such as a teacher who wants to use certain subjects. I'm trying a blog describing selected items culled from 60 or more podcasts each week, thinking it might be a way to make better podcasts easier to locate. No feedback or comment has occurred after 16 entries over 4 months, a short period considering the probability anyone is even looking. I now suspect blog material is read more frequently on smartphones, and my efforts are too wordy for small screens. So we'll begin producing shorthand-type entries. It does a far better job of offering material than an alphabetized list of podcasts on my hard drive. This doesn't address all your TWiV, TWiP, TWIM efforts, especially as their numbers increase, and only a few of them are included in this blog. Anyway, if you contact anyone to suggest an article about use of podcasts in schools, the attached list of 92 RSS feeds with podcast names might be of use. But, Vincent, I guess this is the link that didn't work well for you or something.
Yeah, it's a Word perfect file, I think, WDB. I can't open it, so I can't make it more universal. Alan said MS Works database file, so you have to give me something. I would love to post it for people, but I don't think everyone has that.
So Jim should send something that's a PDF or
PDF
something.
would be good, yeah.
Yeah, okay. So in addition, this link about listening faster might be of use too in light of Kathy Spindler's comment about finding time to hear your episodes. So I went to that link, and it's true. It's got a whole section about how you can adjust podcasts. You run them through some things evidently to make them so you can adjust the speed that you listen to them. And I have to say, sometimes when I'm listening to TWiV, I'll speed it up to 2x, and there's parts of that where that works well. But then there are parts that I have to go back and do that 30 seconds back thing on my iPhone, and sometimes I have to do it 2 or 3 times because you guys can just say one thing and it sends me off on a tangent thinking about something, and then I realize I haven't heard the next 3 minutes of what you said. So listening faster does work. I think there was a listener once who told you he'd gotten caught up and listened to everything at 2x.
Sure.
Wow. Yeah. So anyway, then he has the blog, Media Mining, is included should anyone have an interest in that approach. So that is Jim's blog, the Media Mining blog. And that does it has just a nice compendium of topics that I think people might enjoy seeing, so they can check that out. Suggested improvements are welcome, of course, to include ditching it for some other approach. Oh, I enjoyed your description of daily activities. All time well spent. Regards, Jim.
That was you and I.
That was the show you were on too, yeah.
Right.
Yeah, we finally got around to this, Jim's.
Day in the life, yeah.
I have picked this as a pick of the week, Jim's blog, a while ago, so I would think you would get some traffic there, Jim, but I'm sorry if you're not getting comments.
So Science 360 Radio is looking at that. There are a huge number of podcasts out there.
Oh yeah, and those are all aggregated by NSF. Right? They put that together, and we're there, among others. A lot of people say they find us there, so that's great. I think what Jim is doing is great. I mean, he told me he listens to 60 podcasts a week.
Wow,
it's a lot.
That's a lot.
He must listen at 2x speed or more.
Yeah, so I think it's a good service because I would point people to it. I mean, we've picked it as a pick, and we'll put this link in again. And if you send me a PDF, Jim, we'll put that on as well. I think it's a good thing to do. All right, let's do one more. This is from Stephanie, who writes, Dear TWiVers, I was a bit shocked when a listener wrote in last week and requested that you guys to avoid the subject of evolution in order to avoid offending anti-evolutionary Christians. I understand that in order to reach the largest audience possible, it is important to be politically correct, but is it even possible to discuss papers in the field of virology without understanding viral evolution? Why do viruses become resistant to drugs? Why does a virus tend to be less pathogenic in long-term hosts? How did viruses develop so many ways to subvert host immune defenses? This is a sensitive subject, to be sure, but this subject is something that just about every teacher of biology must face at one point. I look forward to hearing how you guys respond to this issue. You TWiVers are role models for the scientists and educators that do not practice communicating science to a large, diverse audience on a weekly basis.
So I don't think the person who wrote in asked us to avoid evolution, but rather asked us to not sort of trash folks who were religious.
That's right.
Okay? I think we made some borderline disparaging remarks at one point or another about people who, had a religious bent to them. And it wasn't really, we should be polite. And, we took that to heart. But I don't think there was any suggestion that we should not discuss evolution. And if there was such a suggestion, I would ignore it. Because, one of the problems with evolution is that people call it the theory of evolution. I'm sorry. Evolution is a fact. Deal with it.
Well, I teach a lecture on evolution in my course, and I have a slide, which I actually got from Lynn Enquist. And it says viruses are among the best examples of evolution, even for people who don't believe in it. You can't deny it. It happens so quickly. It can happen in an hour, in a lab experiment, that there's no way around it, as opposed to animals where it takes thousands and thousands of years, and you could argue that it doesn't happen because you can't do an experiment to directly show it. So I don't think you can deny that viruses evolve. It's not possible.
Right.
There is mutation and selection, which is the fundamental basis of evolution, and there's no point in denying it. I think Tim Collins, who's a highly religious fellow, the head of NIH.
Francis Collins.
Sorry,
Francis
Collins.
Collins.
Tim Collins is someone else. He's a singer, right? Francis Collins believes in evolution because he sees the scientific basis of it, but he says that doesn't prevent him from having faith. So I think you can reconcile the two.
That's right.
And so that's the answer. It's clearly there, and we can explain it to you in terms of viruses because that's what we do. And there's no reason to believe that mutation and selection is fundamentally any different than any other organism. You know, the rates may be different, but it all happens, and it accounts for diversity. So, what do you think, Kathy? Or do you want to not even get near this?
No, I pretty much agree with what both of you guys have said, that, you know, viruses evolve, other organisms evolve. I think that's a fact. There is this hang-up of it being called a theory of evolution, and if we called it a theory of gravity, people would see them as the same thing. And so I think there's room for both. You know, people that are believers and in Christian faith or other religions, and people that are believers in evolution. So.
Okay, let us do some picks of the week. Let's start with you, Kathy.
Okay, so the homepage I've had on my browser for years is Astronomy Picture of the Day. And I like it because I see it almost every day. There's always a paragraph below with links to other things. Today's is about Jupiter and moons of the Earth. At the very bottom, there's an archive. And so, for instance, you can click on a couple days ago, April 25th, and it's a picture of this meteor over Crater Lake. And then when you mouse over the top of it, the constellations appear. It just has a wide range of things that are just fun to look at for a brief amount of time, or you can follow the links below to other things. And so
it's pretty cool.
Yeah.
So you said you have a program that makes it your desktop automatically? Is that how
it's not my desktop. It's just
on your browser.
On the browser, yeah, the homepage.
So I know people have made programs that pull the photos and make it every day your desktop picture automatically.
That'd be cool.
That's pretty neat. But yeah, I used to look at this all the time. I loved it. I stopped doing it a while ago. I don't know.
Yeah,
why.
somebody who can tell me how to make this my desktop picture or my screen desktop picture would be best. That would be very cool.
I'm
I'd like
sure
that.
among our listeners, someone
somebody will write in it.
will tell us.
Because these are very cool.
That's neat. Yeah, I'm amazed we haven't picked it yet.
Yeah,
it hasn't been, so very good. Thank you.
This Crater Lake, the meteor over Crater Lake, reminds me that when I was growing up in California, we lived in an area that didn't have a lot of city lights, and I used to sleep outside all summer.
Wow.
And I would just go to sleep watching the sky, and you could actually see the Milky Way. And every night there would be meteors. If there's not much light and you're looking up at the sky, you're going to see a few. And it was just terrific.
You would just sleep on a
lounge and a sleeping bag.
A sleeping bag.
All summer long.
This explains everything, Rich.
All right.
That you slept outside.
Yeah. It was great.
Okay. I wanted to say that this picture, the rocks sliding along Death Valley, is really cool.
Yes, it is very cool.
So this is something that's actually known. This rock is moving on its own. Is that right?
I forget. I read that whole story. In fact, I thought you guys had talked about it on TWiV, and that's how I knew about it. But maybe not. It's not really a TWiV topic.
It actually gets blown across the
mud flowing. Dry. Oh yeah, they're caused by how the stones end up in the middle.
Yeah, so that's the February 22nd, 2012.
Cool. All right. Anyway, that's really neat.
Yeah.
Rich, what do you have?
I have a song by an artist named Tom Lehrer, who you may or may not be familiar with.
I do, very much.
I really like this guy. Tom Lehrer is from the 50s and 60s, And he was a mathematician who got his education at Harvard and MIT and taught at both institutions, I believe. But on the side, developed what actually became, I think, his major career, which was songwriting. And he specialized in songs that were, shall we say, black humor, morbid, some of them, satirical, very witty, really a witty guy. And he wrote one song called The Element Song, where he sings the periodic table. And it's absolutely delightful. On YouTube, there are several different renditions of this, all with him singing. The one I chose, and they're animated in different ways to show the elements going by. The one I chose because it actually shows pictures of Tom Lehrer, which I thought was important. And it's just a delightful song. I would like to learn to sing this, but I don't know if I have the stamina. I discovered in looking up Tom Lehrer that he wound up retiring to University of California, Santa Cruz, and taught math there. He taught a course called Math for Tenors, and did gigs, and still lives in the Santa Cruz area. He's in his 80s. Kathy, you had something about the tune for this, because he copped a tune for this one.
Right, it's a parody of the Major General Stanley song from Pirates of Penzance.
Okay.
I am the very model of a modern Major General.
Okay.
Wow.
So,
my son was using this to memorize the periodic table earlier this year. Wow. He would sing this song.
That's terrific.
And Dixon can sing the whole thing, too.
Oh, can he?
Yeah. It's pretty cool.
Oh. Well, I'm just going to have to learn it then.
So this brings into mind the idea. I mean, do you remember some of these movies that have been made to illustrate translation and with people?
Yes.
With people, right?
Mm-hmm.
Yes, there's a YouTube of that.
I picked that
translation
some time
movie.
ago.
Yeah,
that was
Right.
from my generation. Some of my friends, actually. One of the guys who was important in making that was a guy named Fred Dice, or also known as Paolo Dice, who was he was a year ahead of me at Santa Cruz. Went to Stanford, and they made the movie while they were at Stanford.
So shortly after you picked that, someone wrote in and said, you guys should choreograph, something to do with a viral replication cycle. Get some, undergraduate choreographers or dancers to do it.
Right.
Said, yeah, right. So,
and we need a song, too.
So the other day, an undergrad in my course came to see me, and so she's a dance major, and she showed me her CV. I said, hey, do you want to choreograph a virus thing with cells? She looked at me and she goes, I would love to do that.
Great.
excellent.
So we're going to try and work it out. And I said, we'll film it. You can, have it, put it on YouTube. So I said something with virus replication and figure out. She said, yeah, I love choreographing. That's what I'll do. Okay, so it may happen. My pick is something I discovered very recently. How did I discover this? It might have been in my weekly Racaniello Google search. But it is the Sabin Archives at the University of Cincinnati. So at the end of his career, Sabin was at the University of Cincinnati, and they are digitizing his archive. And I was contacted by a woman who is doing this, Stephanie Bricking. So she's a librarian, and I talked to her this morning. She got a grant to do this because she said he saved everything, all his papers and photographs. So they got a grant to digitize part of it. She said it's just too extensive. They digitized 50,000 of his letters, for example, and
Oh my goodness,
they're putting it all on here. And I talked to her this morning because I had actually met him a few times and interacted with him, so she wanted to get my recollections. So this is basically the blog of the libraries, and she writes a lot about this project. And eventually the project will be all online. You can access the archive online if you want to do research or whatever, just to look at it. So this is all his correspondence, photographs, whatever peripheral stuff. And so already she's got an interesting bunch of posts here. There's the commemorative stamp. There's an article. There's an interview with Konstantin Chumakov. His father, Mikhail, was the founder of Soviet virology, and he's the fellow that Sabin went to collaborate with at the Soviet Union. There's a picture here on the blog that you just put up. It's really great. I mean, I was a big fan, and having worked on polio, I thought about his vaccines a lot, but this is just amazing.
What a huge project.
Huge project. And so it will be online for everyone to access.
Terrific.
And when that does, it's going to be amazing because, I got just a couple letters from him, and they're really, really cool. So Stephanie's doing this, and I thought I would pick the blog and have people look at it because it's cool. And then at some point, you'll see if you follow this, it will be public. So this is great. I think this is just the kind of stuff we need to get insight into, how he was thinking and when he was developing the polio vaccine.
Isn't the internet just wonderful?
That's great.
That's just incredible. All this stuff is available for everybody to see.
Yep, it's wonderful.
Terrific.
We have 2 listener picks. One is from Spencer. I would like to propose the book Netter's Infectious Diseases, first edition, as a listener pick of the week. The book, filled with great explanations, is beautifully illustrated by the late Frank Netter, M.D., a pioneer in medical illustration. As a student, one of my classmates at Mount Sinai actually had Dr. Netter as a cadaver.
Ooh. That's weird. I didn't think you were supposed to know who you were working on.
I don't know. I guess it doesn't matter. Dr. Netter taught us so much in life as well as after. The sections on parasitic diseases are particularly well done. So that's from Spencer. Okay, he gave us an Amazon link. I've never heard of that one, but it's pretty interesting. Next one's from Richard. Hi, Vincent and other hosts. I have no silly questions for you this week, but a book that I found interesting, Germs, Genes, and Civilization, How Epidemics Shaped Who We Are Today by David Clark. Thanks again for the series of podcasts that I enjoyed learning from each week. Have you read this book, either of you?
I have not.
I've heard of it, certainly.
That sounds good.
Yeah. I have absolutely heard of it, but I have not, and maybe this will spur me to read it. And that will do it for TWiV 181. We are marching towards 200. You can always find TWiV on iTunes, at the Zune Marketplace, or at twiv.tv. If you're new to TWiV and you like it, go over and please rate it on iTunes. It helps us to stay visible. And that's what we want to do to attract more listeners. We also have an app that you can use to stream the episodes to your iPhone or Android device. You can find that at microbeworld.org/app. And don't forget to send your questions and comments. They're a big part of the show. We love them. TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thanks for joining us again, Kathy.
Thanks for having me. It's a lot of fun.
Really appreciate it. I know it takes a lot of time, and it is greatly appreciated.
I learn a lot.
Good. Rich Condit is at the University of Florida, Gainesville, the southeastern portion of the U.S. Thank you, Rich.
You're quite welcome. Always a good time.
And I'm Vincent Racaniello. You can find me at virology.ws. Kathy, take it away.
You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.